The overall goal of this procedure is to show how to define the structure of factor VIII helically organized on lipid nanotubes by cryo-electron microscopy. This is accomplished by first organizing helically purified factor VIII on lipid nanotubes and collecting cryo-electron microscopy data at liquid nitrogen temperature and low electron doses. Next, the factor VIII membrane-bound structure is calculated by combining single particles and helical 3D reconstruction algorithms. Then, electron tomography data is collected and a 3D reconstruction is generated. Ultimately, the calculated factor VIII lipid nanotube structures are validated by segmenting the 3D volumes and comparing them to the tomography reconstructions.
The demonstrated method can help answer key questions in the field of structural biology and blood coagulation, such as what is the membrane-bound organization of factor VIII and how it relates to its function in coagulation.
The GEM 2100 Lab 6 is equipped with a TEMCON operating system consisting of a computer connected to the electron microscope, a screen with windows reading the vacuum system, illumination system, HT, lens currents, and two panels, left and right, placed on either side of the column. The beam shift X-Y knobs and the multifunction or def stig knobs are on both panels.